The overall goal of this procedure is to track single haploid yeast cells during their entire replicative lifespan. The first step is to make a microfluidic chip containing an array of micropads. Next, the yeast cells are loaded into the microfluidic chip, and because the area underneath the micropads has a height similar to the diameter of a yeast cell, the cells will settle there. Subsequently, a continuous flow of fresh medium is applied over the trapped yeast cells, and the emerging daughter cells are washed away by the flow of medium. Bright field and fluorescence microscopy is used to visualize changes in cell or organelle morphology, protein expression, and protein localization that may occur in aging yeast cells.
The main advantage of this technique over the classical microdissection method is that it's less labor-intensive and that it enables long-term, continuous microscopic images of the entire lifespan of individual yeast cells.
Though this method can provide insight into replicative aging, it can also be used for studies requiring continuous microscopic observation over prolonged periods of time.
Generally, individuals new to this method will struggle, because care needs to be taken at particular steps of the production of the microfluidic chip. In addition, loading of the cells into the microfluidic chip may require some experience.
The silicon wafer mold is produced by soft lithography. For details of its production, please refer to the accompanying protocol text. Cut a piece of tough tag into thin strips of about 0.5 mm by 3 mm with a scalpel. Place the strips carefully on the silicon wafer mold, slightly on top of the channel structure between the inlet channels and the micropad array. Next, cover a glass petri dish.